Right, so this video will be about neurons and signaling. It goes with chapter 37. In particular, it will be chapter 37.1 through chapter 37.3. 37.4, which has to do with neurotransmitters, we will look at next time with selections from chapter 38. So we're going to focus on neurons and signaling, so the structure, the function of neurons, and most importantly, the propagation, I should say the creation and propagation of an action potential. So these are the three important things from this chapter: the structure and function of neurons, and the creation and propagation of an action potential. Alright, so neurons are another name, or the proper name, for the nerve cells that transmit signals between each other and make up your nervous system. So I think most of us are familiar with our nervous system, that innervate feeling for all of our body, our central nervous system, our brain and spinal cord, and peripheral nervous system, all of the motor and sensory neurons that go to our muscles and to our visceral organs and things like that, and they communicate with each other. So this long chain of neurons, nerve cells are kind of what we call neurons in the PNS, or the peripheral nervous system. And they communicate with each other to send information from the outside world to the brain, or from the brain to the outside world. And they communicate via short distances, so between each other, with chemical signals. These are neurotransmitters. And over long distances, this communication is kind of takes on the shape of an electrical signal for our membrane potentials. Right? So in our complex creatures like ourselves, we have a brain, which is kind of the integration part we'll talk about here in a second, the integration portion. But in simpler organisms, or even a simpler extent humans, we have things called ganglia, which are kind of clusters of integration neurons. And they work to help send stimuli externally and internally in a information processing kind of system. And so those of you in, say, AP psychology, we are, at least in biology, we're going to stick to the information processing theory of sensation and of the brain. And in this theory, all of our neurons can be grouped into sensory input, integrative functions, and motor output. And neurons are specialized to handle these functions. So sensory input neurons are very specialized. Integration neurons are specialized in motor output. So sensory neurons, like those you might find to send a signal from your eardrum to your brain, transmit information about external and internal stimuli to your central nervous system. And they're part of the peripheral nervous system, your sensory neurons. So for example, nerves that are in your eardrum that sense the vibrations transmit that signal from the outside world to your brain. Your brain is called interneuron, is an interneural system composed of interneurons, and they're responsible for integrating, processing, and analyzing that sensory input from local circuits that connect sensory neurons to the brain, or ganglia. So this is part of our central nervous system, brain and spinal cord. And they also transmit that new information back out to the motor output neurons, which transmit signals to muscle cells, cause them to contract. So sensory input and motor output neurons are part of the peripheral nervous system, and the integration neurons are brain and spinal cord, part of the central nervous system. they all are composed of neurons, and neurons have a very similar structure independent of which of these three systems they're part of. They kind of look like this. So there is two neurons in this picture. And their cells, they have organelles and nucleus just like all other cells. And this round portion right here that composes most of the cell mass, where the nucleus and the organelles are, is called the cell body. So this is the cell body. You'll see these finger-like or branch-like protrusions from the cell body, and these are dendrites. And dendrites are these branched projections from the cell body that receive signals from other neurons. So here is the dendrites of this neuron. Here's its cell body. It's receiving signals from this neuron. So dendrites receive. You'll also see attached to the cell body, it has one much thicker, longer protrusion, and this is called the axon. And it is where our signal will be transmitted from this neuron to the next one. So our signal is going to travel this way down our axon. And the portion where the axon connects to cell body is called our axon hillock. The junctions are the areas where one axon, the terminal of the axon terminal, meets the dendrites of the second neuron, and these are called the synapses. So synapses is the junction between an axon of one neuron and the dendrites or body of another neuron or cell. And there's actually a space in that synapse, on the synaptic cleft. So there is a slight bit of space in between this cell body, these dendrites, and these axon terminals. And neurotransmitters are the chemical signals that are released from the axon terminals of one cell that carry that message and stimulate the next cell, that travel through the synapse and stimulate this next cell. So messages travel from one neuron to the other through the synapse via neurotransmitters, which are found right there. All right, so basic structure of a neuron. If you're, again, in AP psychology, this is probably a very big review for you. Otherwise, if you haven't taken psychology, you haven't had a little bit of neuroanatomy, this will be new for you. And if you take a little bit of time, most of these structures will become pretty intuitive. All right, so now what we're getting into is how do neurons send signals? And so they propagate electrochemical signals. All right, how is that established and propagated? Well, this electrochemical signal is established due to sodium and potassium ions. So just like in all cells, neurons have an unequal distribution of ions, of charged particles, of charged atoms, between the inside and the outside. And that causes a net potential. And it causes the inside of the cell membrane, inside of the nerve cell, to be slightly negative, and the outside to be slightly positive. And this is what we call a membrane potential. So this resting charge of the membrane due to an unequal distribution of ions. And in neurons, there is a resting potential where the inside of the cell is between negative 60 and negative 80 millivolts. So just normally without your neuron firing, this is the membrane potential. So the inside is negative 60 to negative 80 millivolts negative. Why is the inside negative? Well, it has to do with this guy, the sodium-potassium pump. So the sodium-potassium pump, like we talked about way back in the cell unit, is a membrane pump that pumps out 3 atoms of sodium for every 2 atoms of, or 3 atoms of sodium it pumps out. For every 2 atoms of sodium, I can't read today. So the sodium-potassium pump pumps out 3 atoms of sodium. For every 2 atoms of potassium it pumps in. It's an exchange, a co-transporter. Pumps out 3 atoms of sodium, pumps in 3 atoms of potassium. So it's putting out more positive charge than it's taking in. That results in a high sodium concentration outside of the cell, high potassium concentration inside. But if we're getting rid of 3 positives and only bringing in 2 positives in, we're going to lose net positive charge from the inside of our membrane, and it's going to go outside. So, over time, as this works, the inside of our membrane is going to become slightly negative. We're going to only have about, on the outside of the cell, a 5 millimolar concentration of K+. Very, very small outside, very, very high inside. That doesn't completely make this whole negative potential. In addition, there are ion channels in the cell membrane. There's sodium ion channels and there's potassium ion channels. Potassium ion channels most of the time are open, so they allow for further diffusion of sodium potassium, because there's a high potassium concentration inside the cell, since we're pumping all that potassium into the cell, and a low concentration, 5 millimolar, outside. So even more positive charge in the form of potassium is going to leak back out of the cell. There are not very many sodium channels allowing the sodium chemical gradient to equilibrate. So there's not a lot of sodium channels. So once they're pumped out via the sodium-potassium pump, they're out. But there are potassium channels letting the diffusion of potassium to go back to the outside of the cell to try to equilibrate the concentration. This will result in a buildup of negative charge on the inside, all right? And it will continue to build up until this negative charge is offset by the potassium atoms on the inside. Now they want to equal out their chemical gradient by diffusing this way. However, the overall negative charge inside the cell eventually, since opposites attract, negative overall potential inside, positive charge on the potassium, they will attract each other, and this negative membrane potential will actually prevent more potassium from diffusing outside of the cell. And at that point, we'll reach our resting potential, -60 to -80 millivolts. So it all has to do with the sodium-potassium pump and the fact that potassium ion channels letting some diffuse back to the extracellular fluid. So, now how do we generate an action potential? Well, an action potential is a large depolarization of our membrane. So if we're starting here 60 to 90, any sort of impulse can cause a depolarization or a hyperpolarization. So any stimulus that increases the membrane permeability to potassium. So any kind of stimulation that we do to the membrane that opens up more potassium channels will make our resting potential go even more negative. This is called hyperpolarization, more negative than the resting potential. Right? And this is called hyperpolarization. On the other hand, any stimulation that now increases membrane permeability to sodium, increases the amount of sodium channels opened, will allow sodium to travel through back to the inside of the cell, decreasing the membrane potential, making it more positive, and this is called a depolarization. When the membrane potential of a neuron is reduced. And so this is what the resting potential would be, and if we stimulate the cell and sodium channels open up, it creates influx of sodium, less negative, more positive, a depolarization. When a stimulation of the cell, maybe by oh, I don't know, the influx of neurotransmitters from the previous cell causes a depolarization up to a specific threshold, and it's, around negative 50 millivolts, we reach what's called a threshold value. At this point, sodium channels begin to open. And as sodium channels begin to open, the membrane potential starts to climb. And once it reaches this threshold, positive feedback occurs that as more sodium ion channels open, more sodium ions start coming into the cell, which increases the membrane potential. And increasing the membrane potential will then cause more sodium ion channels to open, which will just synergistically, positive feedback, increase the membrane potential. This is called an action potential. And an action potential is the electric signal that travels through a neuron and is used to send electrical signals. All right? So this is how it works. Kind of at the resting state, we're at our resting potential. The activation gates, sodium channels are closed, pretty much closed, and so are potassium channels. The membrane is at its resting potential. Next, 2. So we're at our resting potential right here. 2. The neuron is stimulated. Depolarization. Stimulus from maybe the axon terminal of the neurotransmitters from a presynaptic neuron causes a depolarization. So stimulation opens and activates the gated ion channels, the sodium ion channels, and they start to have sodium ions come into the cell. So large-scale influx of sodium ions. And the potassium channel are still closed, right here. And if that stimulation is big enough, so it crosses the threshold, this positive feedback will happen in which large influx of sodium will start stimulating more and more and more sodium ion channels to open, and an action potential will be triggered. 3, the rising phase. So depolarization opens and activates most of the sodium channels. So most of the sodium channels are open, lots of positive charge flowing into our cell, our membrane potential is rising. The potassium channels remains closed, and the Na, the sodium influx, makes the inside of the membrane really positive compared to the outside, because we're moving lots of positive charge inside. 4, now we're going to have a falling action potential. Once we reach the highest point, the inactivation of most of the sodium gates will start to happen. Almost right after they open, they'll close again, and there'll be a block of influx of sodium. So our sodium channels are going to close, no more sodium can get in, and that block of influx of sodium opens the potassium channel right here. So reaching this potential opens the potassium channel. Now positive charge is starting to leak out. No more sodium positive can get in, but potassium is starting to leak out down its electrochemical gradient, causing the action potential to fall. 5. It's called the undershot. Now, all of the sodium channel gates are closed, but some of the potassium channels are still open, even though we made it back to the resting potential. So now some sodium or potassium is continuing to go out to make this even more negative than our resting potential is. This is called the undershot. And as these gates close, most potassium channels begin to inactivate and return our membrane to its resting potential. So this undershot happens, it gets reestablished by the sodium-potassium pump to our resting potential. What's important is this period, number 5, the undershot, where it's hyperpolarized, more negative than its resting potential, is called the refractory period. This is the downtime in between different depolarizations. In the refractory period, number 5, a new action potential can't be generated. So even if these ion channels, even if this neuron is stimulated in this resting potential, a new action potential cannot happen. Right? So the second action potential can't be sent during this refractory period. So, kind of last thing, a couple things we're going to look at. Second last thing, how does this action potential be conducted throughout from the whole membrane of the neuron to the next cell? So action potential travels from the cell body down the axon towards the synapse. So it starts in the axon body, and that depolarization happens. We've got our influx of sodium, rising action potential. And as this influx of sodium occurs, it stimulates the membrane adjacent to it. So the depolarization of the action potential spreads to the neighboring region of the cell membrane. So now this neighboring region, now it is stimulated to have its sodium channels open because of the stimulation by the influx, the action potential, the rise in membrane potential of the membrane right next to it. So as that happens, the original part of the membrane starts to repolarize. Sodium channels close, potassium channels open, membrane potential falls. Very important. So as this happens, the action potential travels down the membrane. What's really important is because this is repolarizing right now, even though the adjacent membrane is depolarizing, this section is in its refractory period. Because of that, it can't stimulate a second action potential. This is what prevents the signal going back this way towards the cell body. All of these, the area of the membrane downstream close to the cell body, or upstream close to the cell body, is in the repolarization phase. It prevents the action potential from going this way. It can only go that way. But action potentials are actually very brief, about 2 milliseconds, and hundreds of them can be sent out in just 1 second. The rate of action potential creation is really the only way that your neurons can differentiate types of signals. For example, in your ear, the strength of signal is directly related to the frequency that an action potential is created at. For example, a loud noise that is picked up by the sensory neurons in your ears creates a strong or a very frequent action potential, whereas a soft noise or a not a very loud noise only signals a not so frequent action potential from being created. Lastly, very important, evolutionary adaptation of axons. So the rate of axon potential transmission governs how quickly an animal's neurons can send signals to and from the CNS and respond to a stimuli. Naturally, natural selection would favor faster propagation of action potentials because if an animal's nervous system can propagate action potentials faster, it can respond to changes in its internal and external environment faster and survive. And so, just like a hose or a wire, gauges of wire, a wider or a big axon can increase propagation of axon potential. However, there's a limit to how big these axons can be. It can't get too big, otherwise the functionality of the nervous system is lost. However, vertebrate axons have evolved a structure to get around this, to send action potentials extremely fast, even though they are thin, and that is through the incorporation of myelin sheaths that expedite action potential propagation. So myelin sheaths are these very waxy coatings of the axon. So this is the myelin sheath, and the layers of myelin come in little beads spaced out. And in the CNS, the myelin is created through oligodendrocytes, and in the PNS, Schwann cells create the myelin sheath. So here's a Schwann cell, here's the nucleus, maybe this is a PNS cell, and it wraps around and layers this axon in myelin. And what happens is we get these gaps in between the myelin. These gaps are called nodes of Ranvier, all right, from the person who discovered it. Nodes of Ranvier. And what's interesting is voltage-gated sodium channels are only found at these nodes. This is the only place where depolarization happens. So, because of that, the action potential jumps from node to node. This is called saltatory conduction, and it allows faster propagation of an impulse than just propagating down this neuron without a node. So what happens is the depolarization happens at the nodes, sodium influx, and then the repolarization happens in between the nodes, and then another depolarization, sodium comes in here, and the repolarization happens in between the nodes. So there's not every segment of the membrane becomes depolarized. Depolarization only happens in between the nodes, and then repolarization happens in between the nodes, like this. So it can travel much faster. It can actually jump from node to node, and that conduction via myelin sheath or myelinated neurons can carry this action potential a lot faster and allow the organisms to respond to stimulation much quicker. All right, that's it. We'll be going through that section here on Monday. Let me know if you have any questions.